Good morning, everyone. I'm just going to kick it right off. My name is Monica Veldman. I'm the Director of Global Regulatory Policy at the Alliance for Regenerative Medicine, and welcome to our session on accelerating development through regulatory flexibility, evolving endpoints in gene therapy. So today's discussion is about something that we all wrestle with, which is how to move promising therapies forward when traditional approaches don't fit, and then also how to do that with maintaining rigor and also trust. So first I want to set the landscape a little bit. FDA's Office of Therapeutic Products, OTP, has been testing ways to apply flexibility earlier in development, both in clinical endpoints and also CMC expectations. And there's a growing list of pilot programs and processes and initiatives. So there's, among others, there's RDEP, the Rare Disease Evidence Principles. There's the START program, support for clinical trials, advancing rare disease therapeutics. There's the RDEA, the Rare Disease Endpoint Analysis Initiative, and the CDRP, the CMC Development and Readiness Pilot. And each one probes a different piece of flexibility. There's the right sizing evidence, reusing prior knowledge, and aligning earlier on feasibility versus rigor. And the shared goal here is to make flexibility predictable and not exceptional. And these initiatives sit alongside 3 new FDA draft guidances, which I assume a lot of you have read or heard about at least. There's the Innovative Designs for Clinical Trials for CGT products, which confirms FDA's openness to single-arm and modeling-based approaches. There's the post-approval methods to capture safety and efficacy data for cell and gene therapy products, which signals that some data can come after approval, though the threshold for pre-market evidence is still evolving. And then there's the expedited programs for regenerative medicine therapies for serious conditions, which clarifies how FDA applies fast track and breakthrough and RMAT and priority review and accelerated approval to regenerative medicine therapies with additional considerations for trial design and sponsor FDA interactions. So together, all of these efforts are painting a clear picture of how flexibility can be exercised responsibly, and that's kind of what we're here to unpack. So what's working now and how it kind of breaks down sometimes, and also how we can scale it. So our sort of roadmap for this conversation is we're going to talk about some case studies and examples, and then we'll get into totality of evidence, regulatory tools and flexibility, so what's delivering value within certain pilots like START and CDRP, and then looking ahead. And then we'll reserve a few minutes at the end for audience questions if there are any. And with that, I want to have my panelists introduce ourselves because we have some very important people on stage today. So I'll start with Rachel to my left.
Yeah, good morning, everybody. My name is Rachel McMinn. I'm the founder and CEO of Neurogene. Monica, thank you for the invitation for the panel. Neurogene is a late-stage clinical development biotechnology company focused on developing NGN-401, which is our product for Rett syndrome. Rett syndrome is an absolutely devastating neurodevelopmental disorder, and we'll talk a little bit more about that and how that impacted our ability to leverage START, which is a program that we're going to spend a few minutes on, as Monica mentioned and set the stage for. But we were selected for START one out of 3 gene therapy companies on the CBER side, so it's my pleasure to kind of take you through that. But as we just sort of to set the stage, Rett syndrome, no 2 girls are alike, and I think for any of you in rare disease, this will resonate with you of, how do you design a clinical trial for a rare disease population when individuals are, somewhat different and unique. So I'll go through a lot of that, but before we dive into the details, I'll hand it over to Nick.
Yeah, good morning, everyone. Just thanks to Monica and the organizers, really excited to be on this panel today discussing a really important topic. I'm Nicholas Richardson. I'm a hematologist-oncologist. I recently joined Precision for Medicine as VP of Clinical Development, really to continue to support the development of complex therapeutics, primarily for patients with cancer and rare disease. Prior to joining Precision, I was at the FDA for 9 years. So the last several years, I was the Deputy Director for the Division of Hematologic Malignancies 2, which is the division that oversees products for lymphoma, chronic lymphocytic leukemia, and multiple myeloma. Another aspect of my role that I was fortunate to play at the FDA under the Oncology Center of Excellence since 2019 is actually being a clinical team leader for all the products that were submitted to the Center for Biologics, or CBER, for primarily CAR T cells that were being developed for lymphoma, and then the one in CLL, but also all the ones that are under IND. So again, just really excited to be here and looking forward to a really good conversation. And I'll go ahead and pass it to Louise.
Thanks. Hi, I'm Louise Rodino-Klapac. I'm the president of R&D and technical operations at Sarepta. Sarepta is a genetic medicine company focused on exon skipping, gene therapy, and now siRNA. The focus today will be on our gene therapy programs. And interesting, my career started over 20 years ago in AAV, and the drug I'll talk about today, Elevidys, I began at the bench 20 years ago and still working on it today and continuing the development. So it never stops, even post-approval. And I think that there's a lot of lessons to be learned from that. And there's a lot of optimism around gene therapy, and we'll talk about that and the pathways that we think that we have at FDA to help accelerate therapies for rare disease. The focus today will be around gene therapy for Duchenne, and we'll talk more about endpoints and what it looks like in rare and then ultra rare as well. Turn it to Keith.
Good morning, everybody. My name is Keith Wonnacott. I work for a small AAV gene therapy company, LEXEO Therapeutics. We're developing therapies primarily to target cardiac diseases, rare cardiac diseases. The program I'll talk most about today is our program for Friedreich's ataxia. It's more of a muscular dystrophy, but the primary cause of death is the cardiomyopathy associated with the disease, and we're trying to treat that aspect of the disease. Just for a little bit of background about myself, after graduate school, I went to the FDA, worked for 13 years at the FDA, then worked on I left the FDA to go to Novartis and worked on the Kymriah BLA, and then spent some time at Pfizer before joining LEXEO.
Thanks, everyone. So we'll dive right in. I'm going to start with you, Louise, and we're talking about case studies and real-world examples to kind of ground this conversation. And I'm curious, for DMD, where would you say traditional endpoints maybe fell short, and what alternative approaches or designs proved more meaningful or feasible?
In Duchenne muscular dystrophy, I think we'll probably all end up saying this, it's unique because all of rare is unique. And you don't have a single endpoint that defines the disease like you would in heart failure, for example. And so early on in Duchenne, a lot of the focus was on the 6-minute walk test, and that was prior to gene therapy trials. And I think over time, people saw that that wasn't the ideal endpoint, at least in the populations we were studying. And so we evolved to composite endpoints, where you were looking at a scale of multiple different functions that, like the NSAA, that can combined look at the totality of the disease. Also looking at timed function tests, and then several supportive endpoints. And so in Duchenne, because it's an evolving heterogeneous population, identifying the population to study, the time in which to study, all of these impact selecting an endpoint. And so you end up selecting your primary and your multiple, in our cases, key secondaries. And in many cases, you have early data from natural history studies, you have early data from your trials, but you're also sort of validating the endpoint during your trial. So it's not as well known and studied as it is in larger indications. And many of this evolves over time, and whoever goes first has, the ones that follow have the benefit of learning from that, and that's science. And so in Duchenne, we've learned along the way, and we've implemented what we've learned from one trial to the next. But I think what's important to know is there's not a single endpoint that defines the disease. So I think it's important to pick the right population, the right time. Doing placebo-controlled trials for longer than a year or 18 months is really hard in these diseases where muscular dystrophy, where they're deteriorating every day. And so you don't have the luxury of doing a 2-3 year placebo-controlled trial. So you have to make a decision about how long you're going to study. Sometimes it works out perfectly, and sometimes you learn and you have to look at totality of evidence, what we'll talk about, or implement it in your next trial. So I think there wasn't a perfect answer there. There's still much to be learned, but I think that we've done a good job learning for ourselves and then learning for the field as well.
Yeah.
Yeah, and maybe just to pick up from there on Rett syndrome, so similar types of stories. So when we started our phase 1/2 trial, we didn't know, what's going to happen when you replace this gene. So the gene of interest in Rett syndrome is responsible for 1,000 other genes. These girls have deficits. They're nonverbal, so they don't communicate normally. Their hands don't work. Many of them can't even hold on to an object, let alone point or tap for anything that they want. So they're not communicating with their body. They're not communicating with their mouths as we're used to. They also have substantial gross motor impairment. So some girls can walk, about half of them can walk, but the other half can't walk. And then you layer on top of that, they have a whole host of other disease symptoms such as seizures, difficulty swallowing, maybe difficulty sleeping through the night, depending on the girl, or behavioral issues. So when you run a phase 1/2 trial in a disease that is so complex in a neurodevelopmental disorder like that, the big question is, what is the endpoint? What are you going to see? So of course, in a phase 1/2, your primary endpoint is safety, and then and everything else is exploratory. So in Rett syndrome on the market right now, there is one drug approved, but it doesn't treat the root cause of the disease, and most patients do not benefit from it. So we were able to look back historically and say, okay, that's what they did. They needed a massive trial to see a very, very small effect size. That isn't something that's going to be viable for Rett syndrome. Our product, because we're delivering it to the brain, really important, it's a CNS-mediated disease. We're using an intracerebral ventricular administration, which is accessing the lateral ventricle in the brain. You're not going to be able to blind a study like that. And steroids are not standard of care, and you can't just ethically give kids who are not going to receive treatment steroids to blind it. So we're in a situation where we're in an open label setting, exploring, trying to see what we're going to look for with a prior product that isn't really offering us the roadmap of what will you see. So as we started gathering data, we definitely started to see improvements. So unlike a neurodegenerative disease where you're looking to halt patients where they are, we're looking at the opposite. The girls are halted. They haven't really gained skills. In fact, they've lost a whole bunch of skills, and that's somewhat defining. That is defining of disease, is going through a regression period. And so as we were following these girls over time, we started to see that they were doing better. And that they were starting to gain milestones, being able to climb up or down the stairs, which is highly, highly unusual when we were looking at the natural history database. Being able to feed themselves, using their hands in a meaningful way, holding on to objects, picking up like a blanket, having a fork put in her hand, and being able to use a utensil really for the first time. I mean, so just very incredible things for if you imagine yourself as the parent. But then how do you take that and turn that into an endpoint, right, when no girl is the same? So that was a lot of work by our team to really go in, and thankfully, and I think this is one of the things that is really crucial for anybody working in rare disease, having a natural history database is just so essential. So we were able to go in and mine a database that was sponsored by the NIH. It has over 1,000 patients with Rett syndrome, and do our best to, you're not matching in the same way because there's no sort of specific age you can look at. Like I said, every girl is a little bit different, but really start to ask the question of what are the chances that a girl with Rett syndrome in this age range would be able to gain these types of developmental milestones. So with all of that, we took that to the FDA along with the feedback from clinicians, and we constructed a composite not co-primary, but a composite endpoint where we were looking at a clinician global impression of improvement that had Rett specific anchors, so not just a general one, but something that was rooted in the disease. And then we paired that with a gain in developmental milestones, one of a list of 28 skills that we were able to kind of hone in on and say, gosh, if somebody gained this skill, it's really, really unlikely due to chance. And as a result of that, we were able to articulate and argue for a single arm, baseline controlled, patients their own control trial. So I know that's a lot that I just said, but this is not easy. So anyone who's doing this, I know you all are, this is a labor of love, and you're here because you want to find ways that your treatment is going to work. But we're just really thankful that the FDA, and we'll talk more about this, but that the FDA really is open-minded to innovative clinical trial designs and understanding that requiring just some artificial scale that was developed 20 years ago that was never really meant to be a clinical assessment scale, just holding us to that wouldn't make a lot of sense and wouldn't really measure the value of what a gene therapy is able to bring.
And I'll just quickly add that the beauty of gene therapy is that for many of these rare diseases, we're replacing a gene that's missing. And so the mechanistic function of our therapies is known. And so we've seen with the agency now, if you can provide a mechanistic endpoint, such as gene expression, together with a clinical endpoint, the FDA has been happier with that balance of understanding the mechanism together with seeing a clinical benefit.
And I'll just say, I would love to be able to do that for Rett syndrome, but our protein is not secreted. And there is no biomarker for Rett syndrome. So this was really the big conundrum for us. And again, depending on the disease that you're working on, it's great. And I think LEXEO, and Keith, you should definitely talk about your experience here, because I think it's really valuable for those folks that are working on diseases that's amenable. I think maybe one other point I would make is that the endpoint that we're looking at it's not an accelerated approval strategy. It is a traditional FDA approval mechanism, right? You're looking at gain in function. And for all of you, if you have your regulatory experts, feels, function, survives, those are you kind of have to fit one of those in order to have an endpoint, and we fit into the function aspect. So I think that's something to consider, as you're developing your own strategies for clinical development. Is where do you fit in? If you have something survival, hard endpoint, amazing, fantastic. Yeah. No one's going to argue with that. A lot of these diseases are slower progressing, and that's going to be difficult to do.
Certainly.
Yeah, maybe just a little color. I know we're all talking. So one, this is great. Everybody's eager to share. So I think that speaks to the topic. Just to look at this from a high level, recently joining position and from my time at FDA, FDA is definitely committed to expediting, promising therapies. And I think you're hearing that up here. Victoria's story was incredibly impactful. And so, I think that holds true, and we're seeing a consistency of approach. And I think what you're hearing, again, when you think about endpoints, you want to have a great understanding of where they fit in the causal pathway based on the disease setting and the clinical outcome of interest. And then to your point, does that outcome, one, can you accurately measure it? And then two, does it represent a clinically meaningful benefit? And then does it actually fit the regulatory, evidentiary standard of feels, functions, survives, representative of clinical benefit? And I just wanted to message that, I do think we're seeing consistency of approach, in a tumultuous time, within the agency. And, a really quick example is the initial approval of the auto CAR Ts for third-line plus large B-cell lymphoma. CR rate or complete response rate was the endpoint based on a single-arm trial. And because of the goal of therapy for those patients, complete response was the goal. We had evidence that if you achieved a complete response, that lended itself to improved outcomes of progression-free survival and overall survival. And with the magnitude of the efficacy that was being seen with the initial CAR Ts, that CR rate endpoint was used to support a traditional approval, whereas in other classes of products, that would have routinely been used to support an accelerated approval. And so I think you're hearing today that consistency of approach when there are promising therapies. You know, even yesterday, Myrtle talked about their oligodendrocyte gene therapy, you know, decreasing levels of NAA in patients with Canavan disease. They're also part of the START program. And so in a tumultuous time, I think this consistency of approach is really useful, and you're hearing a lot of those examples today.
Yeah. Well, you answered a question I didn't even ask yet, so thank you for that, Rachel, and all of you, seriously. And I think, I mean, the main takeaway here, I think for me, was this evolution, and you're kind of pioneering this approach that not a lot of other areas of disease kind of have to do. So I think that's kind of what sets gene therapy apart, and it's not easy. So, you know, I want to build on this with how the totality of evidence comes together. So, Nick, I'm going to move to you again. You know, beyond primary and secondary endpoints, you know, what supportive and exploratory measures would help to complete the evidence picture? And I know we talked about minimal residual disease. Is there anything that you want to, you know,
Yeah, thanks, Monica. So maybe just 2 comments, and I'm sure sort of my fellow panel members can add some additional color to this. So one, you know, I think it's a great example, especially in Rett syndrome, where you need to think about a composite endpoint. You know, there's, you know, global manifestations of the disease, and there's not necessarily, you know, one aspect, you know, that can be measured that really sort of informs that benefit risk evaluation. It really needs to encompass all available information. We're talking about a rare disease. And so really having a good understanding, and I think that's where sort of having good natural history data can play a huge role in creation of that composite endpoint, in the sense that you are using a totality of evidence approach with, you know, whether it's a composite co-primary, a primary followed by, you know, descriptive secondary endpoints. All of those information sources are ultimately being used to inform that benefit risk evaluation, so you can meet the evidentiary standards of safety and efficacy. So I think your example, you know, in Rett syndrome is a great example of that. And then my second comment that I just wanted to comment on more broadly, as we've heard talks across the last 2 days, is when we think about cell and gene therapies, and I know this is a rare disease talk, expanding into more common diseases. My time at the FDA, I was fortunate to be involved in the assessment of minimal residual disease negative CR rate, or complete response rate, MRD negative CR, in patients with multiple myeloma. And this was based on a public-private partnership across 2 different organizations who actually took it upon themselves to generate partnerships where they could collect patient-level and trial-level data and run the evaluation to understand the correlation of MRD negative CR to the long-term outcomes of progression-free survival and overall survival. And ultimately, all that information was submitted to the FDA so they could do their own independent analysis, and then it was publicly discussed at an Oncologic Drugs Advisory Committee meeting. And so really I wanted to share that is because it can be a model that people can leverage when they think about the development of novel endpoints as cell and gene therapies move, you know, from the rare disease space to more common diseases, and thinking about how can we validate endpoints and develop novel endpoints that can be used for regulatory decision making. And so I think those lessons learned when you're thinking about more common diseases can be applied within the regulatory framework for rare diseases, and that helps you exercise where that flexibility or that regulatory flexibility may play the biggest role.
Yeah, can I
Please,
jump
yeah.
in? You're making some great points. I think in our case, and Keith brought up this point, we utilized the accelerated approval pathway first, and so we were looking at gene expression and the biomarker initially, and we had to show, obviously, that this was meaningfully likely to provide clinically benefit, and that was initial, and that was predicated based on our functional endpoints. And then in our phase 3, apart from the traditional hierarchy, we included MRI, we included wearable devices, and we were able to show a significant difference using a wearable device measuring stride velocity. And this is a validated endpoint in the EU. It's not yet validated here, but I think that was meaningful in a phase 3 trial. Not only could it be used in Duchenne, but then used widely as an endpoint. It just shows high compliance rate. We also used MRI, which again showed significant results in that year. as well as other biomarkers like creatine kinase. So it's the totality of all of those things, the functional endpoints, the expression, MRI, these are unbiased endpoints that you can't, I'm trying to think of a proper word, that you can't influence. And so I think it's taking everything together, and then you learn at each step of the way, and that's really going to help us all in these fields as far as selecting endpoints. And then also, to your point, as we move into broader indications will help as well.
Yeah. And Keith, I'm also curious from your perspective, when you're considering this sort of supportive data, how would you say sponsors should frame these results so that they can strengthen the case without necessarily, I guess, overstating what they show, but supporting, strengthening?
Yeah, I think that it's really important that the story that you tell is cohesive and understandable. And if you have a single solitary endpoint that's moving in the right direction and everything else is moving in the wrong direction, you're probably going to be in trouble, right? Because the FDA is looking globally. For us, it's heart. It's impacting the heart and cardiac function. And there's lots of ways to look at cardiac function. And if you choose one of those as an endpoint, that's great. But if all the others aren't directionally correct, then it's going to hurt you in the end. And so it's being able to tell the story that mechanistically you have a hypothesis about what should happen, and then when you look across a number of different measures, they're all doing what you would expect them to do in a disease area where what would happen if there were no therapy. And I think that's one of the keys with the agency is really understanding how stable or how well understood the natural history of the disease is, and if you don't understand it really well, having a good comparator arm. There's a lot of talk about using an external control versus a randomized control, and I think what we've seen recently is the agency is open to a number of different ways to control your study, but you need to be able to show that the natural history of the disease is consistent, and so that you can show an impact on the variety of endpoints that you're looking at.
Yeah. And I do, of course, want to talk about the agency, so I'll keep time for that. But Rachel, I just want to give you the chance to also talk about any supportive measures that helped complete the story in Rett syndrome, and how were they presented so that they added to this credibility.
Yeah, I mean, we haven't publicly disclosed a lot of details, but I guess what I would say, generally, when you think about these diseases and they're so complex, whether it's the FDA or it's just down the road, kind of post-commercialization, you should be thinking about what really matters to families at the end of the day, right? What matters to families? And you might get different answers depending on the family that you ask. But in Rett syndrome, we talked about some of the core, cardinal features of communication, hand function, gross motor function, but there's so many other disease manifestations. So if a little girl's, went from choking on drinking a glass of water, and now she can, actively chew soft foods and swallow them without choking. I mean, that is really, really impactful. So I think, without getting into tons of details, I think those are the kinds of things that matter. I wouldn't even call that quality of life. I would call that just real impact in terms of daily living. So just as you're thinking about your endpoints and what you can measure, sometimes there's these fancy scales, and you'll do your best, and FDA might require you to include scales, right, to bridge as much as possible, and you'll do that. But as you're thinking about totality of evidence, just make sure that you're collecting things that actually matter to families.
Thank you. And all of these examples, I think, are only as strong as the agency interaction that kind of come with them. And so I want to bring us to our next topic of regulatory tools and flexibility. And Rachel, I'll go back to you. Of the tools and the pilots that you've engaged with, and you mentioned START, of course, which feature would you say, or features, add the most value, and why?
Yeah, so just to explain START, so this was actually Peter Marks's brainchild, but I'm happy to say that it lives on. And basically the idea behind START was if you could give sponsors regular increased access to the FDA in forms of written communication, email communication, impromptu teleconferences, these just really broad access to the FDA in a regular fashion, could that accelerate development? And the answer that I'm happy to say is unequivocally yes. We were literally a year behind another company that's in the same disease we are, and we caught up. So we're now, both launching registrational trials effectively at the same time. And that's really a testament to START. I don't think we would have been able to do that. In fact, I'm confident we wouldn't have been able to do that without the START program. So I just wanted you guys to have some perspective of what it is. It's regular quarterly meetings. It's a ton of work for us. So our poor regulatory and clinical teams, right? By the time you're done with one briefing package and preparing for one meeting, you're sort of like getting ready to write your next briefing package and ask your next set of questions. But being able to have that back and forth and that iterative dialogue with FDA to really evolve your thinking and hone your ideas, it has been a huge benefit. So we're just really thankful that we were selected for the program. And I think that, to the extent that FDA can, expand this out, I know this is a question of like, well, how realistic is it for FDA to use START? It's not going to be used for everybody. Right. That doesn't make sense. This is not for your standard hypertension product or diabetes where the endpoint is well known. This is really something that's, that's complicated. It's a high unmet medical need and a disease that matters. So I guess what I would say is I think that, the pilot program so far from our perspective has been a huge success. We're allowed to not only talk about clinical development, but really all aspects. So when we think about CMC as being, I'm sure it's not going to escape anyone in this room that we've seen multiple complete response letters from the FDA. CMC is the long pole in the tent. Our head of tech ops is sitting here in the audience and he's nodding his head. Always the long pole in the tent, Ricardo. And so what do you need to do? What investments do you need to make when, and in order to satisfy what bar? And so we're able to really ask those questions on a regular basis, whether there's something that kind of pops up unexpectedly, which happens to all of us. We work in biotech, and that's sort of the nature of working in biotech. But even beyond that is just really planning for a successful BLA outcome.
Yep. And maybe just to add one thing that I think you touched on a lot. When you think about these programs, my time at the FDA, I was fortunate to work with multiple companies that were a part of a lot of the different programs that the FDA offers. I think regardless of programs, it's really about maximizing that opportunity when you do get a touch point with the FDA or even other regulatory agencies across the world. And really what I wanted to convey is if you think about, large pharma is getting a lot of touch points with the FDA. They have multiple products that they're meeting with the FDA on across multiple divisions, multiple therapeutic areas, and so they're synthesizing that information and those learnings and applying it to their clinical development programs. And so when you think about biotech companies, they're not getting as many touch points. And so really it becomes critical to have strategic partners in the planning and the execution of those meetings with the FDA to really make them as optimized and as maximal, maximizing that opportunity to really, continue the progress of your clinical development and, keep you on track for that timeline. Because at the end of the day, the greatest access to a drug is an approved drug. And that's really what we're here to do, to get access to these therapies for patients. And so I think just having an understanding of those touch points with the FDA and other regulatory agencies and really how to maximize those opportunities and having the right team and the right people at the table when you're doing that. And I think the START program is a great example of how that engagement and the frequency of that can really benefit, especially in the rare disease space.
Yeah, and I can add that we're a part of the CDRP pilot, the CMC readiness pilot, and similarly, it's all about increased interaction, right? Increased understanding. Not necessarily increased flexibility. They will tell you that the rules are still the same, right? But it's aligning, there's always subtleties, right? And figuring out how to align with the agency so they're happy and we're happy requires interaction, and these pilot programs give you that opportunity to interact.
Yeah.
And I know that's one of the questions that you have, and just not to steal your thunder, but since I referenced it already, and you're referencing CMC, I think we've seen really some nice changes from the FDA. When you think about a couple of years ago, the feedback was if you didn't run a randomized controlled trial, even if you only had 50 patients in the whole world, it was kind of crazy town, right? You had leadership at FDA saying one thing, and you had the review division saying the exact opposite, and it's like stifling to the industry. You now are seeing a much more, I think, unified leadership, top-down infused view that, look, if we want to be an innovative leader in the world in gene therapy, then we need to think differently about clinical trial design. Amazing, fantastic. Single-arm trials, or at least several of them are being sanctioned by the FDA if you can make the case for it. I'm not saying it's easy. But on the CMC side, that flexibility isn't there. And so then the question just becomes, okay, well, if it's not there, what is it that I need to do in order to get this drug over the finish line? Or maybe, you say you're not comfortable with this, but let me give you some facts on the ground as to why what you're saying is not feasible. And at least being able to have that discussion. So if you can only have that discussion in the context of the BLA, you're sort of out of luck, right? Because there's really not the opportunity to engage with the FDA. They're under a tight timeline, a lot of pressure to really review that application and give an answer. So if you can have those discussions up front and say, like, look, here are the issues that we're facing from a CMC perspective, or things you might not even think are issues, but like, do you guys agree with this? And they come back and they're like, actually, we don't. And then you have to figure out, okay, how do we deal with that? So to me, and I think what you're hearing from Keith is that communication, and Nick as well, like the communication is essential for really understanding where there's misalignment. So as a company, you have time to plan and push back when necessary, and in other cases, shift your plan if it wasn't the right plan that's gonna get you to the finish line.
And maybe just to round it out quickly, I think pushing for live communication, whether it's a telecon or face-to-face, has been incredibly valuable, because I think you can get written feedback and you'll have 5 people sit around the table and interpret a response 5 different ways. And so sometimes we've been in meetings and it's one simple thing, and it was, earth-shattering to our whole development program. So sometimes you may have to push, and they may say, well, if you're satisfied with this, we don't need to meet, but we meet because it's incredibly valuable, even if it's not even the entire time.
Well, you sort of answered the next question I was going to ask. You all are kind of reading my mind as I go along. I know I sent questions in advance, but in addition to that, in addition to what Louise said, what other, clarifications or examples from FDA would most improve predictability across programs, would you guys say? I guess maybe we could do a little round robin.
I just think it would be helpful, and this is maybe more of an audience question, but I think I personally am advocating for town halls where FDA can at least on a sort of non-confidential basis be able to say, hey, we're seeing these types of things, and just kind of provide their position. Right, they did that for a little while, and then they just, with all of the things that are happening at FDA, that is not obviously continued. But I think once they get themselves a little bit settled and they understand what their positions are, I think that would go a long way. Because again, if I'm having an issue, if Neurogene's having an issue, I can't believe we're going to be the first company to face that particular issue. It just doesn't seem likely. And so if that's the case, then how do you get that information out there without saying, hey, we were just talking to Neurogene the other day and we wanted to call them out. So I think there's mechanisms to do it. I think the guidances are helpful. I mean, the innovative clinical trial design guidance that you mentioned. I got into a debate with a Wall Street analyst, and I was like, don't you think this is great? And she's like, well, no, because you already have your trial design. I'm like, but not everybody appreciates, like people are questioning, like, is the FDA on board with a single-arm trial or not? And now they've actually put it down on a piece of paper and codified it. It maybe not as granular as we would want, and you don't know if it's going to apply to you and all of that. But they've put down that they're supportive of it, and you can actually go to them and have that conversation. So I think getting these guidances on paper, even if they're draft, is also helpful, but certainly invite you.
Yeah, I mean, I would say I watched the Duke Margolis replay of the CDRP lessons learned, and the FDA speakers, Ram J. Vatsan in particular, said, here are the most common issues. Potency, process validation, and a couple of others. And they know that the FDA knows the issues. They're the same for all the sponsors. And are they trying? Yes, well, there are guidances. There are recent guidances that came out about accelerating your clinical development. There's a new guide, there was a draft guidance that came out last year about potency. So they're trying. And so that's why this communication becomes so important, is because they recognize the issues, they know the issues. They've had a hard time articulating and helping people get over the issues. And I think it's something that we're learning little by little, and I think we're making progress. There's still a lot of progress to be made, but the good news is they're acknowledging and they're recognizing that there are challenges across the board, and that if they could make a difference.
Nick, any sort of
No, I agree with what
thoughts
people
there?
have mentioned. I will say that, coming from FDA, you really get to touch a ton of different pipelines. And so these common themes do emerge. And so I actually think your thoughts on, how can we actually publicize these common themes and the issues that are sort of crossing therapeutic areas, crossing, divisions, and how can we sort of exercise that knowledge that's being gained, not on an individual sponsor or company basis, but really sort of across, the therapeutic modalities, because you're right, the common things are common. The same issues do come up, and really there are, strategies and ways to think about this that can be tailored towards each individual program. And so I think, having a format and an infrastructure to really sort of promote, that type of learning would be really beneficial for everybody.
Yeah. So I think this communication and this sort of recognizing of common themes and then putting that out there in various forums is likely the steps forward that we need. We don't have a ton of time for questions, but maybe if there's one in the audience, one quick one, we would appreciate it. Okay.
I just wanted to say how impressive it was, how well you guys know your patients as people and understand their daily living. But I was wondering for Rett, how meaningful are patient advocacy groups like RSRT or Reverse Rett? And I was also wondering, in your research, have you been able to find out anything about MECP2 duplication syndrome, since it's kind of like the opposite of Rett for a patient?
Sorry, the acoustics in the room are terrible. So you were asking about MECP2 duplication disorder, and what was the other aspect of your question?
Just how patient advocacy groups might be able to help you.
Oh, how patient advocacy. Yeah, so they're essential to our program, so it's a really good point. Thank you for the question. So as part of our clinical development program, we did interact with a number of the advocacy groups, whether it was, kind of testing out things we wanted to do in our trial, facilitating caregiver market research. So, one of the things we wanted to do was make sure that the milestones that we were assessing actually mattered to families, right? It's like if we measure it and they don't care, then it doesn't matter if we hit the endpoint. So, I think one of the nice things we're seeing with FDA is they're really encouraging sponsors to incorporate caregiver or patient feedback, depending on population. For us, it's caregiver feedback, and we've absolutely been doing that every step of the way, whether it's starting with the phase 1/2, but certainly building the registrational trial incorporated the caregiver voice. And we're going to continue to interact with advocacy as much as possible to make sure that we're really delivering a therapy in a way that resonates with caregivers, because at the end of the day, we're not here for any other reason but to help this community.
Yeah, and I will echo that. We have the Friedreich's Ataxia Research Alliance that is a leading organization for our disease, and they've been involved with us every step of the way. They come to our FDA meetings. They provide feedback on the trial and what is difficult for families and what's working for families and what patients are excited about and what patients are worried about. And in addition to helping with endpoints, as was pointed out by Rachel. So they can, if sponsors allow them, and I think any smart sponsor would, patient organizations can play a really important role.
Yeah, and just to finish out, every protocol that we do is reviewed by our patient advisory committee. It's the same, so it's critical to everything we do.
Well, I want to thank my panelists so much for their expertise and everything that they've contributed, not only to this panel, but to the field. So round of applause, everyone. Thank you.